Welcome to part 1 of lesson 2, cell structure. Think of a factory, like the one in this image. Each part of the factory has a specific role: assembling, transporting, or powering the operation. Similarly, cells are like tiny factories where each structure or organelle plays a crucial role in maintaining life. In this section, we will explore the different parts of a cell and how they work together to keep the factory of life running smoothly. Here are the learning objectives for this lesson. Explain the role of the cell nucleus, identify the cellular structures that make and transport proteins and other macromolecules, distinguish the functions of vacuoles, lysosomes, and the cytoplasm, compare the roles of chloroplast and mitochondria, and describe the role of the cell membrane. These are representations of plant and animal cells and will be used throughout the lesson. As each cellular component is discussed, you will see an image highlighted the component. The nucleus contains nearly all the cell's DNA, and with it, the coded instructions for making proteins and other important molecules. By contrast, prokaryotic cells lack a nucleus. Their DNA containing the same kind of instructions is found in the cytoplasm. The nuclear envelope is a double-layered membrane barrier that protects the DNA. The nuclear pores, these are tiny holes in the nuclear envelope that allow certain components to enter and leave the nucleus. And the chromatin, it is uncoiled DNA plus proteins. The form DNA is in when it is not condensed into a chromosome. And you have the nucleolus, a specific area in the nucleus where ribosomes assembly initiates. Proteins are essential for survival of all living things. They carry out countless critical functions, including the synthesis of other macromolecules such as lipids and carbohydrates. Because of their importance, a significant portion of the cell is devoted to the production and distribution of proteins. Let us look at the process of protein production and distribution step by step. The first step in protein production begins with the ribosomes, which are small particles made of RNA and protein. Ribosomes are found throughout the cytoplasm in all cells. They play a crucial role by assembling proteins. This process is guided by coded instructions from the DNA in the nucleus. These instructions tell the ribosomes the exact sequence of amino acids to assemble into a protein. Once a ribosome begins assembling a protein, the next step depends on where the protein is going. Proteins that are destined for export out of the cell or for specific locations within the cell continue their journey on ribosomes bound to the rough endoplasmic reticulum. The rough endoplasmic reticulum is called rough because it is studded with ribosomes, which give it a bumpy appearance. Here, proteins are fed into the RER for further processing. This step is essential to prepare proteins for their final destination. In contrast, the smooth endoplasmic reticulum or smooth ER lacks ribosomes and is involved in the synthesis of lipids and detoxification processes. The next step involves packaging and transport. Newly assembled proteins are packed into vesicles, which are small membrane-bound structures. These vesicles carry the proteins from the rough endoplasmic reticulum to the Golgi apparatus, the cell's distribution center. The Golgi apparatus modifies, sorts, and packages proteins for their final destinations. Some proteins will leave the cell, while others will be directed to specific locations within the cell. In summary, protein production and distribution is a carefully coordinated process involving several cellular structures, such as ribosome that assemble the proteins, the rough endoplasmic reticulum that processes proteins destined for specific location or export, and vesicles that transport proteins to the Golgi apparatus where they are prepared for their final roles. This complex system ensures that proteins necessary for life are produced, processed, and distributed efficiently, reflecting the remarkable organization within the cell. Now that we have followed the protein as it is produced by the ribosomes and processed by the rough endoplasmic reticulum, let us continue tracing its journey. The fourth part is the vesicle that carried the protein from the rough endoplasmic reticulum. It's the same one shown here. Its role is to transport the protein to the Golgi apparatus, the next stop in this process. Once the vesicles reach the Golgi apparatus, it fuses with the Golgi membrane. This is where the protein undergoes further modification. The Golgi apparatus is like a post office for the cell. It modifies, sorts, and packages proteins and other materials that originated in the endoplasmic reticulum. These modifications ensure the protein is fully functional and ready for its final destination. The fifth one is the next step in this process involves the protein being shipped out of the Golgi apparatus. The modified protein is packaged into a new vesicle which will deliver it to its final destination. These destinations vary. Some vesicles transport proteins within the cell, delivering them to organelles like lysosomes. Others export proteins out of the cell, such as those needed for communication between cells or for repair processes. This efficient system ensures that proteins and other materials are distributed where they are needed, allowing the cell to function properly and contribute to the organism's overall health and growth. In summary, vesicles play a key role in protein transport, acting as carriers that deliver materials between organelles like the rough endoplasmic reticulum and the Golgi apparatus, and finally to their ultimate destination. This process is yet another example of the highly organized and precise functioning of cells. Now that we have gone step by step through the process of protein production and distribution, let's take a moment to review and summarize the entire process as shown in this complete image. This slide represents the full sequence we just discussed, from the ribosomes to the final destinations of the proteins. Let us check your understanding of the processes with a few questions. Please pause the video and try to answer the questions on your own, and play again to see the correct answer. Where are proteins assembled? Proteins are assembled on ribosomes. What can you infer about a cell that is packed with more than the typical number of ribosomes? A cell with an unusually high number of ribosomes likely produces more proteins than a typical cell. For example, cells in the pancreas produce a large quantity of digestive enzymes and therefore contain many ribosomes. Where is the synthesis of membrane proteins completed? The synthesis of membrane proteins is completed in the endoplasmic reticulum or ER, specifically in the rough endoplasmic reticulum. How are proteins transported to the Golgi apparatus? Proteins are transported to the Golgi apparatus in vesicles, which are small membrane-bound structures. What happens to proteins leaving the Golgi apparatus? Proteins leaving the Golgi apparatus are either sent out of the cell or delivered to specific locations within the cytoplasm to carry out their functions. This ends Lesson 2, Part 1.